Good day, students. This is Ma'am Quaj. Welcome to Clinical Chemistry 1 laboratory subject. Today, we will be discussing everything about carbohydrates, particularly its biochemistry and its clinical significance. But before I will proceed to my lecture proper, allow me to humbly ask the understanding of everyone that this lecture might consume more than an hour. But since this is a laboratory subject and it has one unit, and one unit corresponds to 3 hours for the laboratory subject. So I guess what I have said earlier is just acceptable. And in addition to that, since we are in the new curriculum, some of the topics that are supposedly to be discussed in Clinical Chemistry 3 will be discussed today. So moving on, here are the learning outcomes for today's lecture. The students will be able to describe the carbohydrates according to composition, classification, and chemical properties, discuss the metabolism of carbohydrates in the body, explain the mode of actions of different hormones in the maintenance of blood glucose levels, and discuss the different processes involved in the maintenance of normal blood glucose level. As you can see, the learning outcomes of numbers 1 and 2 are learned already in your previous subject, and that is biochemistry. So in my first few slides, this is just an overview or a review of what you have learned from your previous subject. We all know that carbohydrates are the major food source and energy of the body and are stored as glycogen primarily in the liver and in the muscles. Carbohydrates are compounds containing carbon, hydrogen, and oxygen. And this carbon to hydrogen and to oxygen ratio is 1:2:1. And carbohydrates have this general empirical formula of CnH2ON. And some books may write this as CH2ON, where N is the number of carbon atoms attached to its molecule. Now, glucose is the most important carbohydrate in the body, and it is considered as monosaccharide. And in this case, glucose has this general empirical formula or chemical formula of C6H12O6. Carbohydrates have two functional groups, which are the carbonyl group, from which the carbon is double bonded to oxygen, and another functional group, which is the hydroxyl group. Now, carbohydrates are said to be either the hydrates of aldehyde or ketone compounds with multiple hydroxyl groups. That's the reason they are said to be polyhydroxy, because of the presence of several hydroxyl groups attached to the structure of these molecules. Now, as you can see, we have two different sugars on this slide. We have the aldose and ketose. Both of them have suffix of "-ose", meaning to say it refers to sugar molecule. Now, in the case of the aldose, it has this aldehyde compound which is attached to its structure. Aldehyde in the sense that it has carbonyl group and hydrogen atom attached to the carbon atom. While for the ketose, it has this ketone group attached to its structure. It has the carbonyl group and two carbon atoms attached to the carbon atom present in the middle. Each sugar molecule is held together by what we call as the glycosidic bond or glycosidic linkage. And this glycosidic bond is a type of covalent bond that joins together a sugar molecule or a carbohydrate molecule with another group. And this group may be or may not be a carbohydrate. So on this slide, as you can see, we have this sugar molecule named as lactose, and it is also called as the galactosyl beta 1,4-glucose. Now, why it is named as such? So we will discuss one by one. Now, lactose is a disaccharide, meaning to say it is joined together by two monosaccharides such as the glucose and galactose. These glucose and galactose have the same number of carbon atoms, which is 6. And as you've noticed under structures, they have multiple hydroxyl groups attached to it. Now, it is said to be 1,4 because of the connection or the reaction between the carbon number 4 of glucose and carbon number 1 of galactose. So to better explain this glycosidic bond that held together this sugar molecule, let's have another slide. Okay, on this slide, we have here the lactose. It is a disaccharide which consists of two monosaccharides such as the glucose and the galactose. Glucose and galactose have the same 6 number of carbon atoms on their structure. So to start counting the number of carbon atoms on these molecules, to start here. For the glucose, this is carbon number 1, 2, 3, 4, 5, and 6. Now, this CH2OH is called as the methyl hydroxy group. Now, for the galactose, let's start counting on this portion. This is carbon number 1, 2, 3, 4, 5, and 6. Now, lactose is a result of the condensation reaction between the hydroxyl oxygen atom present in carbon number 4 of glucose and carbon number 1, which is said to be the anomeric carbon of the galactose. Now, when you say condensation reaction, that refers to a process or a type of reaction wherein the water is a byproduct. Okay, that is when you form two monosaccharides or two molecules together, you will release or you will form water as its byproduct. Now, if you want to separate, if you want to break them apart, from a larger molecule to a smaller molecule, you will use water to do so. Now, as I mentioned a while ago that the carbon number 1 for the galactose is said to be the anomeric carbon. Take note of this class. In the cyclic structure, the carbon that is present in the carbonyl group of the linear or straight chain structure of the molecule is said to be anomeric carbon. Now, in this case, the carbon number 1 of the galactose is the anomeric carbon. Okay, now that is the reason why this lactose is called as or has this 1,4 glycosidic bond. Now, how about for the beta? What is the difference between beta and alpha? Now, the difference is simply the position of the OH group on their cyclic structure. Now, when you say beta, that refers to the position of OH group above the ring of its structure, of its cyclic structure. Now, aside from that, the OH or the hydroxyl group that is placed in carbon number 1 is on the same side of the CH2OH. So that is called as beta glucose. Now, on the other side, on the left side, we have another or other sugar molecule, and that is the alpha D glucose. It is named as alpha because the OH here is placed below the ring of its structure. And aside from that, the OH group or the hydroxyl group is placed not on the same side with the CH2. So I hope you understand why the lactose is called as the galactosyl beta 1,4 glycosidic bond. To sum it up, we have here the 1,4 because of the positions or the connection between the fourth carbon atom of glucose and the carbon number 1 of the galactose. It is named as beta because of the position of the OH group, which is placed above the ring and on the same side of the CH2OH. Now, I mentioned a while ago that the carbon number 1 present in the glucose, as what has been presented in the previous slide, is considered as the anomeric carbon. It is considered as anomeric carbon when it is changed into cyclic structure. So this is now the linear or straight chain structure or the Fischer projection of the glucose. Okay, now we have here carbon number 1, 2, 3, 4, 5, and 6, and it has the aldehyde group. And in the aldehyde group, it has the carbonyl group from which the carbon is double bonded to oxygen and it has the hydrogen attached to it. And this is the cyclic structure of the glucose. Okay, this is the cyclic structure of the glucose. And since in the cyclic structure, the carbon atom that is present in the carbonyl group in the linear structure is considered as the anomeric carbon. So in this case, the carbon number 1 is the anomeric carbon for this molecule. So 1, 2, 3, 4, 5, and 6. Okay. Galactose and glucose have the same aldehyde compound attached to their structure. They are also called as aldohexose. Why? Because they have the same number of carbon atoms, which is 6, and they have this aldehyde compound attached to their structure. For how about fructose? Same with galactose and glucose, they are said to be monosaccharides. Actually, they are the most common monosaccharides. But the difference is that though fructose consists of the same number of carbon atoms, they have fructose has the ketone compound attached to its structure. Then we start counting the number of carbon atoms here. Carbon number 1, 2, 3, 4, 5, and 6. As you've noticed in the structure, we have the carbonyl group in the carbon number 2. Now, as I mentioned, the carbon atom that is present in the carbonyl group in their structure, in their straight chain structure or linear structure, is said to be the anomeric carbon. Okay, so this carbon number 2 here in the fructofuranose is the anomeric carbon. Now let's start classifying carbohydrates based on the following: size of the base of carbon chain, the location and type of the carbonyl functional group, the number of sugar units, and the stereochemistry of the compound. Now let's start the classification of carbohydrates based on the number of sugar units. Here is the classification of carbohydrates based on the number of sugar units. We have the monosaccharide, the oligosaccharide, and the polysaccharide. Now, for the monosaccharide, it is named as such because it consists of a single sugar unit, and it cannot be any more further hydrolyzed because it is already in its simplest form. And examples of which we have the aldoses, we have here the glucose and galactose, and for the ketoses, we have the fructose as an example. Now, for the oligosaccharide, it consists of 2 to 10 number of sugar units. Now, for the oligosaccharide, it has this disaccharide, which consists of 2 sugar units, and examples of which we have the maltose, the lactose, and the sucrose. These are the 3 most common disaccharides. And another, we have the trisaccharide, and we have the tetrasaccharide. Now, for the polysaccharide, it is named as such because it consists of many or several number of sugar units. Examples, Examples, we have the starch, the dextrin, the glycogen, the cellulose, and the inulin. Now, these polysaccharides are said to be complex carbohydrates. Now, for the another classification of the carbohydrates based on the number of carbon atoms, we have here the trioses, which correspond to 3 carbon atoms. We have the tetroses, 4 carbon atoms. The pentoses, 5 carbon atoms. Hexose have 6 carbon atoms, and the heptoses, we have 7 carbon atoms. Here are the different cyclic ring structures of the different sugar molecules. We have the glucose, the fructose, and the sucrose. Here, the glucose is considered also as the pyranose ring structure, while the fructose has this furanose ring structure. Okay? Now, for the sucrose, it consists of 2 monosaccharides such as the glucose and the fructose. Have you noticed the glycosidic bond that is present between these 2 monosaccharides? Now, this glycosidic bond is formed between the carbon number 2 of the fructose, which is the anomeric carbon of the fructose, and the carbon number 1, which is the anomeric carbon for the glucose. Now, some scientists consider this as one of the reasons why sucrose is a non-reducing sugar because of the glycosidic bond that is formed between the same, okay? They are joined between the two anomeric carbons of the different monosaccharides. I think I have addressed this already. The classification of carbohydrates based on the number of sugar units. We have the monosaccharides, the disaccharides, the oligosaccharides, and the polysaccharides. Now, for the disaccharides, which has 2 monosaccharides joined together, we have this sucrose, consists of the glucose and the fructose. We have the maltose consists of the glucose and the glucose, and the lactose consists of the glucose and the galactose. Examples for the polysaccharides are the following: the starch, the glycogen, and the cellulose. Now for the starch, it consists of the polymers of amylose and amylopectin. Now consider the difference between the structures of amylose and the amylopectin. Now, when you say polysaccharide, this consists of a long chain of sugar units. Now, for the amylose, it has this unbranched long chain of sugar units, while for the amylopectin, it consists of the long chain of sugar units which happen to be branched. So there is branching of these sugar molecules that are connected together to form a polysaccharide. Now, another basis for the classification of carbohydrates is the location and type of carbonyl functional group. We have here the ketone and the aldehyde. Now, aldose consists of the aldehyde compound, while the ketose consists of the ketone compound. Now, the examples for the aldoses are the glyceraldehyde, ribose, glucose, and galactose, while for the ketoses, examples are the following: the dihydroxyacetone, the ribulose, and the fructose. Now, consider the structures of glucose, galactose, and fructose, which are the three most common monosaccharides. They have the same number of carbon atoms, and the same elements or atoms present, and the carbon, hydrogen, and oxygen, but they differ in their properties as well as the spatial arrangement. Okay? And aside from that, the fructose has the ketone group compared to the aldehyde compounds present in glucose and galactose. Now, the last basis for the classification of the carbohydrates is the stereochemistry. Now, there are 5 important types of isomerism found with glucose. Number 1, the DNL isomerism. Second, the pyranose and furanose ring structures. Third, the alpha and beta anomers. Next, the epimers, and the aldose and ketose isomerism. What do you mean by isomerism? When you say isomerism, it is the phenomenon in which more than one compound have the same chemical formula but different chemical structures. And these chemical compounds that have identical chemical formula with but differ in their properties and in their arrangement of atoms in the molecules are called isomers. Now, in this case, we have the D and L isomerism. It has the orientation of H and OH groups and specifically found at carbon number 5 in the case of glucose and the carbon atom adjacent to the terminal primary ROH carbon. Now, we have here the example of D-glucose and L-glucose. Please notice the position of the OH. Okay, OH found in D-glucose and L-glucose as well. Now here, in the D-glucose, the OH is located in the penultimate carbon to the right, while in L-glucose, the OH is located in the penultimate carbon to the left. What do you mean by penultimate? It is called as such in the sense that the OH is found in the last chiral carbon molecule that is present in the sugar molecule. Now, in this case, the last chiral carbon atom is found in the carbon number 5. Okay? When you say chiral carbon atom, it is the carbon atom which possess 4 different types of atoms or 4 different groups of atoms. So, we have here 4 chiral carbon atoms that are present in glucose molecule. Okay, now how about for the L-glucose? The L-glucose molecules still have the same number of chiral carbons with respect to the D-glucose, and it is also specifically found in carbon number 5, but the difference is the OH is found in the penultimate carbon to the left. That is why it is L-glucose.
When you say chiral carbon, definitely it corresponds to the number of isomerism. So since D and L glucose are isomers with each other, so how many numbers of isomers do this glucose have? So since the glucose have 4 chiral carbon atoms, with respect to the formula of the number of isomers, which is 2 to the n, and n corresponds to the number of chiral carbon atoms present in the molecule, we have, in this case, glucose has 4 chiral carbon atoms, so 2 to the 4 corresponds to 16 isomers. So definitely glucose have 16 isomers. Now, another example is the glyceraldehyde, which is the smallest carbohydrate. We have here the isomers of glyceraldehyde and L-glyceraldehyde. Now, same explanation with the previous example, that the OH here is found at the right side of the penultimate carbon, while the L-glyceraldehyde, the OH here is located in the left side. Okay, so the chiral carbon here, or the last chiral carbon atom that is present in the D-glyceraldehyde, is at carbon number 2. Okay, so next slide. We have here the pyranose and the furanose. As I've mentioned in the previous slide, when you say pyranose, it corresponds to the 6-membered ring. Take note of this. It is a 6-membered ring, while the furanose corresponds to the 5-membered ring. Okay, I will not go into detail about this because you have learned that in the previous biochemistry subject. Another, we have the alpha and beta anomers. I guess I have addressed this also already. When you say alpha, that means that at carbon number 1, the OH group, or the OH rather, is found uh, below the ring of the molecule, below the cyclic ring of the said molecule, and also not on the same side with the CH2OH. While when you say beta, the OH at carbon number 1 of the sugar molecule is found above the ring and also with the same side with the CH2OH. Now, these are all the Howard projections that represent the cyclic sugars as having essentially planar rings with the OH at the anomeric carbon 1. So, as I mentioned, alpha has OH below the ring and beta OH above the ring. Another type of this that will explain the stereochemistry of the carbohydrates is through the epimerism. When you say epimerism, it is the stereoisomerism if two monosaccharides differ from each other in their configuration around a single specific carbon other than the anomeric carbon or anomeric atom. Now, in this case, compare the structure in the D-mannose and D-glucose as well as the D-glucose compared to D-galactose. Now, in the sense of in the case of the mannose, here, compare with the structure formula of D-glucose, they have almost the same. Okay, they're almost the same in terms of the structure formula, but differ only in the carbon number 2. Okay, differ only in the carbon number 2. See? So in this case, D-mannose is epimer at carbon number 2 with respect to D-glucose. How about the D-glucose in comparison with D-galactose? They have almost the same structural formula, but they differ only in carbon number 4. Take a look at this. Okay, so the D-galactose here is epimer at carbon number 4 with respect to D-glucose. Now, carbohydrates are said to be the major fuel of energy for the cells of the body in order for them to function well and, of course, to survive. Okay? And one of the major goal or role of the carbohydrates is to release energy in the form of adenosine triphosphate or ATP. Now, what happens to the food that we ingested? Okay? Through the mouth. So, through the mouth, these complex polysaccharides or complex carbohydrates will be digested via the salivary amylase as well as the pancreatic amylase into dextrin and also to the different disaccharides such as the maltose, lactose, and sucrose. Now, these disaccharides will be correspondingly, okay, broken down into its simplest form, and that is the monosaccharide. With respect to maltose, this maltose will be reacted by its enzyme named as maltase to be broken down into glucose and glucose, while lactose will be acted upon by the lactase. An enzymatic reaction will take place in order to break it down to galactose and glucose, while the sucrose, which consists of which is acted upon by the enzyme sucrase, will be broken down into its simplest form, such as the fructose and glucose. Now, these are the simplest form of the carbohydrates. Okay, these are monosaccharides. Now, upon this digestion, of course, this will be taken up by the gut and traveled into the liver via the portal hepatic venous blood supply. And this will go into the circulation, of course, and this will be absorbed by the cells of the body. Now, what happens to the cell when they absorb this carbohydrate, the simplest form of sugar? On this slide, we have different stages of catabolism, and these are stage 1, digestion and hydrolysis, stage 2, degradation and some oxidation to smaller molecules, stage 3 is the oxidation to carbon dioxide, water, and energy for ATP synthesis. Now, these are just the general overview of the stages of catabolism. When you speak of catabolism, of course, we cannot forget the word anabolism. When you say catabolism, that refers to the breaking up of things into smaller molecules. And anabolism refers to the building up of things into larger molecules. As you can see, these are the different biomolecules in the body. We have the proteins, the polysaccharides, and the lipids. They have different pathways, of course. And when you say proteins, this should be broken down into its building blocks of units, and that is the amino acids, its simplest form. And amino acids, through the process of transamination, will undergo the different pathways here, as you can see, while the lipids will be broken down into its simplest form, and that is fatty acids or glycerol, from which the fatty acids, via the beta oxidation process, will be converted to acetyl coenzyme A in order to enter this pathway. And how about the polysaccharides? So we will focus the discussion on the polysaccharides, since the polysaccharides are the complex carbohydrates. So these polysaccharides need to be broken down into incipients form, and that is monosaccharide, such as the glucose, fructose, and galactose. Glucose is the most important carbohydrate. Now, this glucose will enter different pathways, 3 metabolic pathways. These are the glycolysis, the citric acid cycle, and of course, the oxidative phosphorylation cycle. To start with the glycolysis, I know you have learned a lot from your previous biochemistry subject about these different metabolic pathways. Now, you still remember that the glycolysis produces these products such as the ATP, which is the energy currency of the cell, also named as adenosine triphosphate, and also the pyruvate. So aside from the pyruvate, it produces the NADH. Now, glycolysis produces, as I mentioned, pyruvate. And this pyruvate, which is produced here in the glycolytic pathway that occurs in the cytosol of the cell, pyruvate will be converted to acetyl coenzyme A in order to enter the citric acid cycle, which occurs in the mitochondrion organelle of the cell. Take note that the glycolysis is an aerobic process, while the citric acid cycle is a cellular respiration that needs oxygen in order for the process to take place. So upon the conversion of pyruvate to acetyl coenzyme A, this will enter into the citric acid cycle, or also termed as the tricarboxylic acid cycle or the Krebs cycle. It is termed as the tricarboxylic acid cycle because of the presence of 3 carboxyl groups that are present in the citric acid. Now, just to remember that in the citric acid cycle or the Krebs cycle, there are 8 enzymatic reactions that take place. in the first enzymatic reaction, the acetyl coenzyme A it will combine with the oxaloacetic acid in order to produce a citric acid. And this citric acid will now consist of 3 carboxyl groups. And this is now parang siya ang umpisa or siya ang gagamitin in order for the next enzymatic reactions to take place. And by the way, I forgot to mention the other term for the glycolysis is the Enden-Meyerhof, which corresponds to the name of the discoverers. To continue, in the citric acid cycle, it produces, of course, the major goal of this metabolic pathway is to produce an energy needed by the cell. Aside from that, it will produce the NADH and the FADH2. These are strong energy carrier molecules. But to compare between the two, NADH is a very good electron donor from which the electron is at its high level, and the FADH2 is not so good donor electron because the energy is at lower level. Okay, and this NADH can produce 3 ATP rather, while the FADH2 can produce 2 ATP, and that is in the process or in the pathway of oxidative phosphorylation, which consists of 2 important components such as the electron transport chain, and the chemiosmosis. So this is the general overview of the cellular respiration or the metabolic pathways from which the glucose can be used up in the form of energy. We have the glycolysis, the conversion of pyruvate to acetyl coenzyme A, and this acetyl coenzyme A will enter the citric acid cycle, and this NADH and FADH2 will be needed by the last or the final cellular respiration, and that is the oxidative phosphorylation consists of these 2 components, electron transport and the chemiosmosis. This is where the bulk of the ATP is produced. Okay. Next, this is the electron transport chain. I will not thoroughly discuss the process, the whole process, but I will just give you a short review of this electron transport chain. Now, as I mentioned, the oxidative phosphorylation is the final stage for the cellular respiration from which there is a proton gradient formation in the electron transport chain, and which in turn to production of the ATP in the ATP synthase via the chemiosmosis. So take note of that. Now, in the electron transport chain, it consists of proteins, several proteins, and it consists also of 4 complexes. We have the complex 1, 2, 3, and 4. And we have also here the ATP synthase. Now, this NADH, since it is a very good donor electron, which has the presence of electron at its high energy level, it can directly enter to the complex 1. But the FADH2, which has the electron that has at lower energy level, so it cannot directly enter the complex 1, but instead it will enter to the complex 2. So the first stage in the ATC cycle is the delivery of the electrons, and the next stage will be the transfer or the travel of these electrons from one complex to another. Okay, now as you can see from the matrix of the mitochondria, the hydrogen is pumped out into the intermembrane space, and this can cause the change in the concentration gradient. Now the oxygen as the last electron acceptor will be split into 2 and form water. Okay, and will split into 2 to form water. That is how important the oxygen is. But because without the oxygen, of course, this ATP production will not be possible. Now, these hydrogen ions at his low gradient will enter the ATP synthase, and this is where the ATP production takes place via the process called as the chemiosmosis. This is just the same explanation with the previous slide, so I will not go through with this. Do you still remember what this cycle is? Yes, this is indeed a Cori cycle. Now, I remember my previous students in my biochemistry subject in the previous year level, I told them that there is no such thing as building up of things that will occur at the same time with the breaking up of things. Why are you breaking things and at the same time you're building up things at the same time? It doesn't make any sense. But in this case, the Cori cycle, there is the building up of things and the breaking up of things which occur at the same time, but take note of different cell. And these cells include the muscle cell and the liver cell. Now, glucose is the major food source of energy. And while for example, if there is a man that is doing vigorously exercise or a vigorous exercise, a quick workout, of course, the oxygen will be insufficient. But since the glycolysis in the muscle does not need oxygen in order for the glycolysis to take place, the glucose here will be converted to one of its products, and that is pyruvate. Now, this pyruvate will be converted to acetyl coenzyme in order to proceed to its next cycle, and that is Krebs cycle. But how come or how this pyruvate will proceed to its next cycle, and that is the Krebs cycle, if there is no enough oxygen that is present in the muscle cell? Okay? Now, of course, it will not take place. So what will happen now? This pyruvate will be converted to what we call as the lactate. And this lactate is toxic to the body. Since it is toxic, we don't want to build up lactate in the body. And this lactate will travel in the blood circulation, and this will be taken up in the liver. Liver because it is said to be the detoxifying organ of the body. And in the liver, the lactate will be converted to pyruvate. And from that pyruvate, since in the liver, there is the presence of oxygen and it is where the cellular respiration can take place, and the lactate here will now be converted to pyruvate and eventually will be converted to acetyl coenzyme and enter now the Krebs cycle. And eventually another cycle will take place. Okay, so this is just a cycle. So take note that in the muscle cell, glycolysis takes place, and in the liver cell, gluconeogenesis takes place, in which there is the conversion of glucose from non-carbohydrate sources such as the amino acids and other proteins, and in this case, that is the lactate. So these are the different pathways in glucose metabolism. We have the glycolysis, glycogenesis, glycogenolysis, and gluconeogenesis. When you say glycolysis, of course, that is the metabolism of glucose to pyruvate or lactate for production of energy. Glycogenesis, there is the conversion of glucose to glycogen for storage. And this time, the glucose is stored as glycogen primarily in the liver and in the muscle for storage, so that when there is a need for us to use this stored energy, we can break this glucose for in order to produce or to release energy. And that is a process called glycogenolysis, the breakdown of glycogen to glucose for use as energy in the future, especially when there is stress and or vigorous workout done in the body. Another is the gluconeogenesis, which is the formation of glucose 6-phosphate from non-carbohydrate sources such as proteins and fats. Take note that the glucose 6-phosphate is parang siya doon nag-umpisa yung pathway to proceed to the HMP or the hexose monophosphate pathway, from which it is very useful for the production of the nucleic acids or for the process for the nucleic acids. And take note also that the glucose 6-phosphate is the first substance that is produced in the glycolysis pathway by the action of the hexokinase. So the next topic will be discussed in the next slide.